Thank you, Chichu, for this. So, my talk will be mostly about molecular genetic diagnosis. This is one branch of the three branches of human genetic and medical genetics. One branch was addressed first in the morning, biochemical genetics, by Dr. Wani, and then cytogenetics by Prof. Ravindran, and then now I'm coming into this molecular genetics, because the substance that are studied are different. For biochemical genetics, you're studying enzymes, metabolites, those kind of things, and then for cytogenetics, the substance studied are chromosomes, and then for me, the molecular genetic, the substance studied are the DNA itself. So where it all happens. Next. Then where it all happen. So this is where it all happens. In the cells, we have trillions of cells, and practically human body is a building blocks of cells. And in every cells, we have the so-called core of cells, nucleus. And nucleus is where the DNA, most DNA are located. We have, like previous talks, perhaps you already know, we have DNAs in the nuclear of cell, and also we have DNA in the mitochondria. But most of the genetic informations are located there in the cell. Next slide. So we have 46 of these, 23 pairs of chromosomes. So chromosomes basically are how the DNA are packed. If you stretch the DNA, like Prof. Sawi just mentioned, it's 2 meter long. 2 meter long. And then that 2 meter long DNA will have to be packed into a super tiny 6 micrometer, 0.006 millimeter of nucleus. That is comparable to probably having a string from Kota Baru to Machang. It's probably around 40 kilometers, and then pack it into a tennis ball. So how it is packed, that's how chromosome play its role. Yeah? And then the work of the work of molecular geneticists is to break that package and then identify the sequence of the DNA in that packaging. Next, so I perhaps can start with some scenario of where molecular genetic diagnosis play its role. So it can play its role in the monogenic disorders, where abnormality of certain genes will manifest disease. Example here first is tuberous sclerosis complex. It is an inherited tumor which is inherited in the from parents to the offspring to the kids. So in this disease, we have only 1 or 2 genes which are involved, and then once mutation happen in that gene, and then the disease will manifest. But the problem with this disorder is that it encompasses a huge segment of DNA of genome, 2 genes, each with hundreds of thousands of kilobases, and no particular hotspot where the mutations can be found, and no particular type of mutations of how we can identify. Particular type of mutations is important, because then we can determine the type of test that we want to conduct for identifying patients. So big regions, no specific target mutations, and this is mainly for diagnostics. But then another scenario will be spinal muscular atrophy. This is also similar with tuberous sclerosis complex. You have mutations in a certain gene, and then once the mutations occur, then you have the disease manifest. Right? But the difference is that with spinal muscular atrophy, we deal only with 1 gene, and then we have common mutations. where 95% of the patients have that mutation, right? And that type of mutations is commonly detected with a simple test, a simple molecular genetic test, the so-called targeted variant test, or the test name RFLP. Next slide. And then yesterday we also talked about pharmacogenetics and pharmacogenomics. We heard some patients develop serious and deadly adverse events because of consuming certain drugs. Example that I take here is carbamazepine, which is one of the anti-epileptic drug, and then SJS is Stevens-Johnson syndrome. Some of us may have heard that. Stevens-Johnson syndrome. When you have Stevens-Johnson syndrome, so you have all the mucosal part of your body removed. So this can be deadly. This can be very, very deadly. One gene, which is HLA, and then specific variant, HLA B1502, and then a simple detection method. But when somebody have that variant, not necessarily that person will develop Stevens-Johnson. In fact, only 7% of the patient that have the variant of the genetic of the gene will develop Stevens-Johnson. But the negative predictive value is absolute, meaning that if you don't have that variation, you will be safe. You will be safe. If you have the variations, you have a possibility to have Stevens-Johnson, although it's very, very small, but deadly, right? But if you don't have it, if you don't have the genetic variation, you will be safe. None of the individual without genetic variation for SJS will develop Stevens-Johnson. So this is important in terms of ruling out deadly adverse events. And then breast cancer. There are two genes, BRCA1 and BRCA2, so-called BRCA1 and BRCA2. We will have more detailed information about cancer genetics from Dr. Sarina later in the evening, and then specific set of variants. Simple detection method as well, but then this is increasing the risk until 8 to 10 times. The recent example about this is probably many of us already hear about Angelina Jolie removing her breast. Upon knowing that she carry genetic mutations that may predispose her to breast cancer, and she has a strong family history for that. So this is specific target mutations, and then the purpose of this is risk prediction. Risk prediction. So next slide. So next slide. Yeah, and the next slide. All right. So these are the list of the why we need to conduct molecular genetic diagnosis and screening, and we have to bear in mind there are two terms here. One is the cause, another one is the risk. This will have something to do with the so-called penetrance of the mutation. So when the penetrance is absolute, is 100%, it means that you have the mutation and then you manifest the disease. Like the first two examples, tuberous sclerosis and spinal muscular atrophy. One which is more common in our population is thalassemia, for example. You have the mutation in beta globin or alpha globin gene, and then you have thalassemia, alpha or beta thalassemia. And then another terminology is with moderate or low penetrance, which we talk about risk. You have the mutation or the variant, means that you have increased risk. Now, how many times you increase the risk, that will depend on the relationship between the genetics and that particular disorders. Later in these presentations, if I have more times, I can give one scenario of communications between genetic epidemiologists and clinicians about that. Next. So this is the aim. This is particularly for molecular genetic diagnosis. This is the ultimate aim: identification of pathological or variant DNA changes. DNA changes can be many types, so-called missense, nonsense, deletion, insertion, any type. So basically, identification of DNA changes, right? We see from cytogenetic workup, it's identification of the number or the structural changes in chromosomes. And we see in the biochemical genetics, it is the fluctuation of the amount of metabolites, of certain metabolites in the body. So with molecular genetics, we can see the identification of changes in the DNA. There are basically 4 different, if I can lump it into 4 different methodologies, we can have these 4. Targeted variant detection. We already know what variation or what DNA changes that we want to target, and then we do that specific test. Currently, this number 1 made up the most of the tests that we have. And then the, number 2 is sequencing. Sanger sequencing, identification of sequence of DNA, urutan DNA, jujukan DNA itu, for a very limited segment of genome, the segment of which we know very much linked with the disease. And then number 3 is microarray. Microarray is similar to targeted variant detection, but if in targeted variant detection you only see 1 or 2 or 3 variation, but in microarray you have thousands, tens of thousands of variations. You have large number of it, right? And then number 4 is the next generation sequencing. Different with Sanger sequencing. In Sanger sequencing, you can only detect extremely limited segment of genome, but with next generation sequencing, you can have more. You can even sequence the whole DNA. Or most applications for this is that to sequence the whole gene. One gene cannot be sequenced by only Sanger sequencing. Probably one tiny portion of certain gene can be sequenced by Sanger sequencing, but not the whole gene. But the whole gene can be sequenced easily by next generation sequencing. Then next generation sequencing will be very beneficial for case number 1 that I mentioned, with big genes, no common mutations, no particular position in the genes which are prone to mutation. So we and then each of the gene encompass tens of thousands of DNA. So next generation sequencing is 1. So these are the disadvantage problems and advantages with the different methodologies that we have. So each have their own advantage and disadvantages. That depends mostly on what do you want to identify. Right? For spinal muscular atrophy, we know that 95% of patients have that particular mutations. So we prefer targeted variant detection. We aim to target that particular mutations, which cover all 95% of the patients, right? And then we have Sanger sequencing, microarray, and next-gen sequencing. The cost, as the technology is getting newer and newer, of course the cost is getting higher and higher. But the so-called high throughputness is much increased. The coverage of that test is also increasing. This data I got from United States, of course. Proportion of technologies that are used in the molecular genetics. So 69% of the molecular genetic problems are identified by identifying DNA. And then out of that, apparently more than half of the tests used targeted variations detection already. And then almost half, about 44%, use limited sequencing, Sanger sequencing. And then only about 3% 3%, and then 1.5% use the most current technology. But this figure in the next 5, 10, 15 years might change, I guess. Maybe as the cost for microarray and the cost for next-gen sequencing is decreasing, then more tests will be done using that, right? Like I can illustrate in 98 from 98 to 2003, where the Human Genome Project ran. It took them $100 million to sequence the whole DNA of one person. The whole DNA of one person. Now, how much to sequence that? That cost probably only $3,000. So it's very much decreasing, and the cost keeps decreasing. And how it decreases very fast. It doesn't follow the economic theory. Right? It's decreasing very fast. So, and then what to consider when a doctor or even a patient wants to have a genetic test? What considerations should be taken into account, especially with regards to molecular genetic tests? So we have 3 things at least. We have four things that I'm going to discuss later in slides. But the main considerations now, there are 3 things. First is analytic validity. Second is clinical validity, and then the third is clinical utility. Analytic validity means how much chance of that particular test can identify the changes that it is supposed to identify. Sensitivity and specificity of the test. For example, test RFLP for spinal muscular atrophy. I repeatedly use it because we have the test in Human Genome Center. Use a certain kind of test. It says it will test the deletion of certain part of the gene, right? It claims to be able to identify that changes. And then using that particular change, how much possibility that that test really truly identified that if it is positive, if it says positive, then how much specific that, how much sensitive that, how much the chance of that particular test really identify that it is really positive or it is really negative. So we're talking about that particular test. And then this covers like analytic sensitivity of the test itself, specificity of the test itself, and then quality control along the way test is carried out. And then the assay robustness. And then once it is cleared, usually in the United States, it is all labs that that are doing genetic tests. They have the so-called certification from organization named CLIA, C-L-I-A. That's an organization to certify the analytic validity of genetic tests. And then once we cleared with analytic validity of that certain test, then we go up to clinical validity. Let's say now we trust the test, all right? It turned up positive or negative. Then example for in case of SJS, Stevens-Johnson syndrome that we already discussed earlier. If you have that particular patient positive for that certain genetic mutation, then how much chance of him or her having Stevens-Johnson syndrome? Or in that particular case, how much chance of somebody with negative results without any particular genetic mutations will develop Stevens-Johnson? Right? So this is what we call clinical specificity, clinical sensitivity, and then also now it's widespread also in the community and also in the United States. In the United States, when you go to, I was surprised. I did some shopping to Walmart It's a few, I think 2 years ago, and then I found a box saying paternity testing. It's sold everywhere, just in Walmart. Paternity testing. What you do is you do a buccal swab, or you collect any like hair or any remainings, and then you send it to a certain lab. Lab will come back to you with results. And what's also overwhelming now is if you go to 23andMe, you can search in Google 23andMe. The website offers genetic testing for a wide range of common disorders, like diabetes, like cancers. Okay? So later, probably, if we still have time, we can see somebody approached a doctor with 23andMe test results, saying that the test say that she has 25% increased risk of having diabetes, while the common populations have 21% of risk having diabetes. So there is only 4% of increased risk. How much that information have benefits to the patient? Right? So that has to be taken into account, really. And then the last one is the clinical utility. The clinical utility is something that after you know the results, okay, the test is robust, okay, you trust the test, and then the test itself can identify how much risk you will have to you will have the disease if you get the mutation, and then so what? What can doctors do about that? Any preventions can be done, right? In terms of this is another example. I use again Angelina Jolie example. So she really don't want to have breast cancer. So she found that there is now the so-called prophylactic mastectomy, right? Removal of breast before the cancer manifests, in order to prevent the cancer to manifest. Right? I think it was just a few days after that, I also read in newspaper another one having mutations of prostate cancer, and then having his prostate removed. That one, I don't know. We have to have much discussions on the clinical utility of that. Right? All right. Then next, the fourth one, the fourth utility that we have not really put that into the scheme of genetic tests is this personal utility. Most example of this is probably on a disease that you cannot really prevent that from occurring. You know that somebody have that mutation, and the risk for that person of having that mutations to develop the disease is high, and then nothing you can do in the future. Some patients do find it beneficial for them to know the information prior, in advance. Example here is Alzheimer's disease. It helped them to prepare for the future despite lack of intervention options. Next. This is this originally used the example of DMD, Duchenne muscular dystrophy. So, no, we know that we see the clinical symptoms of that patients match clearly to Duchenne, and then we see that the test show positive. So what? Nothing. No, as of now, no curative treatment for that particular disorder. Okay. Yes. No? And then there's no curative treatment, but in fact, actually, patients and families, they are satisfied with the information given to them, because most patients usually they are when we have that kind of disorders. Then there are queries with them. What kind of disorders is this? Right? So if we have that test saying that, okay, this is Duchenne, so we know they know understand. And then another thing is that if you don't have proper diagnosis, none, either the doctors or the patients, will know where the patients will land, what will become the patients, what will be the prognosis. But if the diagnosis can be established, then the patients understand, all right, the chance for life is probably until early adulthood or teenagers. So understand where the prognosis may land. But then how personal it is. I put up an example here. A father requested BRCA1, BRCA2 mutations for cancer for his 8-year-old daughter. The mother and the auntie died of breast cancer. The kid, the daughter doesn't have authority to say yes or no, is it? Whether she wants to know it or not. But the father deadly needs to know, right? Right, because she really needs to know that. So we need to see. Next slide. Yeah. So basically, these are the schemes. When you have most likely, if you have the frequency of certain genetic mutations very high in the population, and that frequency will increase also risk, then the penetrance is low usually. So the chance of that particular genetic mutations of giving disease is very low usually. While if you have the frequency of the genetic mutations is very high in the population, very low in the population, usually the penetrance is very high. It's the case, the two examples perhaps in the high penetrance will be diseases like thalassemia, spinal muscular atrophy, Duchenne muscular dystrophy. And then the example in the lower part may be like cardiovascular disorders, hypertension, diabetes mellitus, like that. Right. Okay, next. I don't think I have times for this because this will take like about 12 minutes. But we will anyway upload this to YouTube if you want to have the clear. This is a video of scenario between conversations between genetic epidemiologists and a physician who consulted queries of her patients about a genetic test from 23andMe that discuss about 25% increased risk in the diabetes mellitus. We will upload this to YouTube as well. Then you might want to see it when we have uploaded. You can submit your emails if you want to have the video or link to the YouTube. Right. I guess thank you very much. The upper one is the thank you from Jawa. It's I'm from Jawa. It's thank you in Bahasa Jawa. Thank you very much. So I guess I just move on to questions now, which can be directed to either me or Prof. Ravindran, or perhaps also Dr. Wani, if any of us can answer that. Yep. Nobody? Nobody? If nobody, perhaps we can use the time to see the video, is it? Right. You know.
In this next scenario, a patient comes to you, the physician, with a report from a direct-to-consumer genetic testing company showing that she's at increased risk of diabetes and wants you to help her interpret the report. Before you talk to the patient, you consult with a medical geneticist about these test results. So, Bryce, what can I do for you?
I'm back with another question. This patient of mine went to 23andMe and ordered genetic testing done that she was hoping would help her predict her risk of type 2 diabetes.
Okay.
And so she brought the report to me, which I have right here. And it says that she has a 25% lifetime risk of type 2 diabetes compared to 21%. And I'm just asking you to fill in the black box for me. How did they go from her blood sample to this result?
Right. Well, if you take a look at the report, which I see here, again, like you mentioned, she's got a 25% increased risk over the general population, which is a 21% risk. Basically, the question is, how did they come up with this 25% risk, right? Well, if you look down in the lower left, you'll see that that is a composite risk score that they derived from information about 11 different single nucleotide polymorphisms. So think of these as 11 independent risk factors. And what they did here in this case was each of these risk factors has an associated odds ratio that was derived from some genome-wide association study, for example. What they did was they took each of those odds ratios and they multiplied them together.
Okay.
And so they come up with this composite odds ratio. Then they had to convert that to a relative risk, which is a slightly different but more accurate measure of your risk, for example. And based on that relative risk, then they multiply that by the average population risk to come up with your 25% risk. Does that make sense?
That makes sense. I do have a few questions about the details.
Okay.
So you said these are 11 independent risk factors. Do we know that? Do we know that all these genetic changes are actually independent of each other?
We don't know that. That's a big assumption that went into this. They were all discovered individually, but nobody's really put them all together in one model and said that they are indeed independent. And nobody knows, do they interact with each other? Would you get a different risk estimate if you put them all together in some other way? You very well might.
Okay. And so similarly, do we know if you can really just multiply them like that?
Right. That is one way of doing it. It may or may not reflect what's going on.
Okay.
But it is the way that they did that.
And then my other question, it sounds like to calculate her risk and to compare her risk, they needed to have an average population risk.
Right.
Where did that come from?
Those estimates usually come from these large epidemiological studies. You've heard of the Framingham Heart Study. Studies like that where they survey a bunch of people in the general population, they measure their level of risk of disease, and that's what they use as the general average population risk.
And is there a way for me to know if the sort of racial ethnic makeup of this average population matches my patients?
That's a very insightful question because, the genetic basis of these diseases may very well differ by ethnicity, and these genetic markers may behave differently depending on your ethnic background. And similarly, you need to make sure that whoever you're comparing it to, for example, the average population, is the same ethnic background as the person that you're talking about.
Okay. So a little more complicated, I guess.
A little more.
I'm also a little bit confused. With 11 genetic markers, why is the change in risk from the average so small?
Well, the change in risk is so small because genetics is not the only factor contributing to type 2 diabetes.
Okay.
We know that for type 2 diabetes, only about 25% of the risk is due to genes.
Wow.
And when you think about that subset of disease that is heritable, how many genes are involved? Well, we don't know. So far, we've identified about 39 genes that contribute to that. And even with those 39, if you take them all together, they only explain about a third of that genetic component. So looking at just these 11 is not capturing the whole picture.
Okay, so I'm not going to be able to get a report someday that says your patient will not get type 2 diabetes ever, or definitely.
Probably not. Probably you got a ways to go.
Okay.
And even if you had all of the genes, even if you knew all of the genes, that's not the whole picture. That's the point.
Okay. And then I was also, wondering. I heard in the news that the patients aren't going to be able to get this testing from this company, at least for the moment. What's the issue there? Why can't people get their genes sequenced if they want?
Well, that's a very complicated issue and very controversial right now. I mean, there are a number of things that the FDA is taking issue with. I think first and foremost that they had requested 23andMe to work with them in order to get FDA clearance for these tests ahead of time, and that conversation was not continued. 23andMe decided not to continue it for whatever reason. Some of the issues that they had were related to the clinical validity of these tests. That's one thing. How well do these tests actually predict risk of diabetes, for example? And can you give accurate information to patients? Do these tests do what you say they're going to do? And then there are also issues with the fact that these were going direct to consumer. There was no physician there to help interpret the results.
I see. I can tell the tension between wanting patients to have access to their own genes and yet have that access be informed and verified.
Yeah, where do you sit on that? I'm just curious.
I'm a little on the fence. I think that I can imagine a person having a right to the sequence of their own DNA. It is theirs in a very unique way. But I do think that if you're going to tell someone how their health is going to change, that there should be some form of oversight or regulation. What would you advise someone then? I know that 23andMe isn't at the moment offering health-based screening, but these tests aren't going to go away, and they're only going to get more complicated. What advice would you give me, if a patient comes to me and asks if they should do this test?
Right. So first and foremost, I think you're doing the right thing coming and talking to me, and hopefully physicians will become more educated about genetic testing in general and be able to field these questions themselves one day, right? So you need to consider whether the test has clinical validity, whether the test has clinical utility, how much it costs, whether it's covered by insurance. There are a whole range of factors that you need to consider before deciding whether you're even thinking about ordering this test. But now you've also got these situations where patients are getting this information on their own and bringing it directly to you. So you need to be able to interpret those results as well. Okay.
Well, thank you very much for going through this report with me. Are there any other things I should know about this particular panel of gene tests that might help me with this patient?
Well, I guess one word of caution, and that is, as you saw, the methods that are used to come up with these composite risk scores are, they're depending on a lot of different factors. They're depending on the number of genetic variants you include in your cumulative risk score. They depend on the average population that you use. I just want to show you the results of this one study. Before 23andMe, there were actually 3 different companies that offered this direct-to-consumer genetic testing, 23andMe, Decode, and Navigenics. And this is a really interesting paper where a group compared the risk estimates they got from these 3 companies, and they asked what went into coming up with these risk estimates and how did they vary across the testing companies. And you can see the average population risk used for each of these companies was pretty similar, except for a couple of diseases like age-related macular degeneration and celiac disease. You can see the average risk in the population that they used varied somewhat. They also varied in the number of different SNPs that they included in their risk estimate. So you can see for the different companies, sometimes twice as many risk markers were used in some companies versus others. So those are two of the ways in which these testing companies differed. And then when you actually look at the results, what did they tell you? Did they tell you you were at increased risk of disease, decreased risk of disease, or no change? And you ask about concordance between those 3 companies. You can see that the rate of concordance, if you add up all of these numbers, for example, you can see what the overall rate of concordance is. Either they all 3 reported a decreased risk, all 3 increased, or all 3 neutral. You can see that the rate of concordance varies depending on the test. From a low of like 33% concordance for prostate cancer risk to a high of about 89%, which was for the
celiac disease?
for celiac disease.
Yeah, I see that.
Right?
Okay.
So that's one thing. What's worrying is when you have situations where the 3 companies, where either 2 of the companies said increased risk and the other said decreased risk, or they all 3 had very different results. And you can see that happened some of the time as well. So those are the things that you have to worry about. Different companies using different numbers of SNPs, different average population risks, and so on, and coming up with these very different risk estimates.
So it sounds like there's a lot of work to do. It sounds like someone will have to come up with a set of SNPs and a way of analyzing them, and then actually follow patients and see what the predictive value is, and then also follow those patients beyond that point to see if they had any health benefits from knowing ahead of time. And that's a lot of work.
That is a lot of work, and when you think about, sometimes these discoveries are made and people are very anxious to get them into the clinic to help patients, and I completely understand that. You don't want to have to wait 10, 20 years to run all of these tests to show that they have some clinical utility. You want to be able to have this information now. So there's a real balance, though, between having good information and having the innovation and new technologies out there in clinical practice. And you're going to see that on the front lines as a physician.
Okay. Well, thank you so much.
You're welcome.
The following are the key concepts that we hope you got from this second case scenario. First of all, multi-marker genetic risk profiles, or panels, should be interpreted with extreme caution. Genes identified to date poorly explain the genetic underpinnings of many complex diseases. Several resources exist to find objective information and guidance on genetic tests. Genetic testing should not distract from modifiable risk factors such as diet, lifestyle, and medication use. And finally, there are two sides for the controversy over direct-to-consumer genetic testing.
Morning. Still allow probably if there is any one of you who wants to raise questions. No? All right. So we have our tea break after this for probably until 11 before we go to the next session.